Can you talk about how do you think about developing a vaccine? Then they talk about these phases. What are the phases? What do they mean, please?
Okay, so we'll start from the beginning. Let's suppose you think that you want to make a messenger RNA vaccine. So messenger RNA is just a little piece of genetic material that is the code for how to make proteins. We all make messenger RNA in our bodies because we all make proteins in every cell of our body. So let's suppose that's your idea. So I'm going to take messenger RNA and I'm going to inject it into cells, and then hopefully that messenger RNA will be translated to a protein, in this case, the SARS-CoV-2 spike protein, because I know that antibodies directed against that protein can in theory protect against transmission because it'll protect the virus from binding the cells. Because now you have your idea. I want to make an mRNA vaccine. The first thing you do is try and create an animal model for disease. So whether it's mice or monkeys or ferrets or hamsters, you inoculate the animal with that virus that you're interested in, and hopefully they'll get the symptoms that people get. Then you try out your vaccine to see if it works to protect the animal and what part of the immune response that's generated in that animal seems to be protected. Those are called proof of concepts. Then you go to people. You do phase 1 studies where you try and make sure you have the right dose that induces that immune response that you think is protected from what you learned in your experimental animal model studies. Then you go to phase 2, which doesn't involve just 20 to 100 people like dose 1. It involves hundreds of people to make sure that this now vaccine that you have that's being given at a specific dose is consistently induces that immune response and is safe. It doesn't cause a common serious side effect. Then you go to phase 3 studies to prove that your vaccine works. So you give the vaccine to tens of thousands of people, then you send them out into the world to see whether or not those who got the vaccine are more likely to be protected against disease than those who weren't.
Let's talk specifically about COVID-19 vaccines and public perception. And so clearly we've never developed vaccines this fast. So I want you to talk a little bit about the speed of the vaccine and how we were able to come up with really good end stage candidates for this vaccine.
How are we able to do it so quickly? As distinct from previously, where the fastest vaccine that was ever developed, to my knowledge, was the mumps vaccine, which took roughly 4 years. The reason is that the United States government took the risk out of it for vaccine makers. What they did was they put $24 billion into this program and said, okay, we'll pay for phase 3 trials, which cost hundreds of millions of dollars. And most importantly, we'll pay to mass produce this vaccine without knowing yet whether it works or it's safe. So you had that sort of overlap. I mean, while you were doing the phase 3 trials, you were already mass producing the vaccine. No company would ever take that kind of risk, because what the government was saying is, okay, we'll wait to see whether or not it works and is safe. And if it works and it's safe, then we'll put it out there. But if it doesn't work, or it's not safe, or both, we are going to throw away hundreds of millions of doses. That's why it's so fast.
So why is it so hard then to develop a vaccine? And with what's happening nowadays, are there new techniques and technologies that we're learning that will help us for vaccines in the future?
That's a great question. What we're seeing now are the so-called genetic vaccines. In the past, what you would do is you would say, take a virus, grow it up, and inactivate it with a chemical. That's the way that we made the polio vaccine, or the hepatitis A vaccine, or the rabies vaccine, whole killed virus. Another thing you could do is take a virus and weaken it so that it can reproduce itself in your body enough to induce an immune response, but not enough to cause disease. The so-called live attenuated viral vaccine approach. That's the way we make the measles vaccine, the mumps vaccine, the rubella vaccine, the chickenpox vaccine. Or you can really use so-called recombinant DNA technology. So this is relatively recent, just in the last roughly 20 or 30 years, where you actually just give that one protein you're interested in. And usually it's the protein like the SARS-CoV-2 protein, and this is another strategy that's being used to make this vaccine. You give the person the protein you're interested in to induce an immune response. That's the way we make the hepatitis B vaccine. That's the way we make the human papillomavirus vaccine. In each case, you're really giving either the virus or part of the virus to induce an immune response. What's so different now with all these approaches, the so-called vectored virus approaches or mRNA approaches or DNA approaches, is what you're really giving the person, you're giving them the gene that codes for that protein. So they therefore make in this case, the coronavirus spike protein, and they make antibodies to that spike protein. These approaches are novel. There's virtually no or very limited commercial experiences with them for any other pathogen. So you're right. What will we learn from this? I mean, if the mRNA vaccines are as good as they look, and certainly the sort of top line data, the press release data looks great, will we be able to make a human immunodeficiency virus vaccine, an AIDS vaccine? Will we be able to make a malaria vaccine or a better tuberculosis vaccine? We'll see. I mean, it is exciting, and it certainly opens up a new field.
So how do you personally help patients? Because you're a medical doctor as well, which is great. So you've given your life to heal people and take care of our loved ones. So again, thank you for that. How do you personally help patients with their concerns around vaccines? And also, where should the average Joe and Jane person look for credible data to help assuage their very legitimate skepticism and questions on certain issues?
I think when people are concerned about vaccines, and it's reasonable to be concerned, I think you should be concerned about anything you put into your body, especially a biological agent that's being injected into your muscle or under your skin. Well, the first thing you do is find out what's their concern. And invariably there are data to address that concern. We have a website at vaccine.chop.edu, which is part of our vaccine education, where we have tried to answer every question we have ever been asked about vaccines. And then we have a vaccine safety kind of repository where with each of those questions, we'll have a series of studies which you can link to. And then we have like a 2 to 3 sentence description of that, because we are trying to answer questions people have. And it's not just us, obviously, the CDC, the American Academy of Pediatrics, the Mayo Clinic. There are a lot of different groups out there that try and put out good, credible, accurate, up-to-date scientific information about vaccines to answer questions people have.